Good afternoon. My name is Nathaniel. I'm a founder of Nivien Therapeutics, and we are eliminating the shields that protect cancer from existing treatments. The vast majority of people with cancer are prescribed chemotherapy as the front line of care. However, in many cancers, chemo remains both ineffective and highly toxic, with terrible side effects. Nivien is developing the first small molecules that can reformulate with a wide range of chemotherapies to create new products in every major cancer that have higher effectiveness and fewer side effects. Recent advances in biology have made it possible to understand cancer at the molecular level. We now know specific proteins responsible for the failure of chemotherapy. For example, drug metabolizing enzymes like cytidine deaminase break chemotherapies down into an inactive form. Multi-drug efflux transporters like the ABC family of proteins find chemo within cancer cells and kick them out, rendering chemo ineffective and poisoning healthy surrounding tissue. Collectively, drug resistance proteins like these are responsible for the failure of dozens of chemotherapies. But past efforts to target this resistance have failed because they have only been able to go after individual proteins, leaving others to pick up the slack. Our team from Harvard and MIT have identified the first powerful upstream regulator of these downstream resistance proteins. This target, called Hippo, represents a unique opportunity to eliminate the barriers that protect cancer from treatment. When Hippo is active, drug resistance protein levels are high and chemotherapies are ineffective. When Hippo is inactive, drug resistance protein levels are low and chemotherapy is able to more easily enter and kill cancer cells at much lower doses, meaning more effective treatments and fewer side effects for patients. We set out to prove this at IndieBio using shRNA to inactivate Hippo in drug-resistant cancer cells. Here, we saw 50 to 70% reductions in levels of key drug-metabolizing enzymes and drug efflux transporters. This translated into dramatic reductions in the viability of pancreatic cancer cells that had previously resisted every available treatment. What's special about Hippo is that it's the only target in cancer demonstrated to play this key regulatory role on so many downstream resistance proteins. As a result of this target validation and this unique therapeutic potential, we have begun developing the first potent inhibitors of Hippo activity. Here, one of our small molecules is able to inactivate Hippo at nanomolar concentrations, a key measure of drug-like potency. Our aim is to reformulate small molecules like this candidate with chemotherapies to produce new products in every major cancer that have higher effectiveness and fewer side effects. This enables a unique business model. Branded chemotherapies generate hundreds of millions of dollars in annual revenue while covered by 20 years of patent exclusivity. However, when those patents expire, revenue plunges as prices fall to meet generic competition. That is where Nivien comes in. Nivien reformulations are not only a better product for patients with higher effectiveness and fewer side effects, they also restart the 20-year patent exclusivity of the new reformulated products, which belong exclusively to Nivien, and mean that we can recapture hundreds of millions of dollars in otherwise lost revenue for every drug with which we reformulate a Nivien molecule. And because Hippo is a regulator of proteins responsible for the failure of dozens of chemotherapies, we can do more than 40 of these reformulations to go after a total addressable market of over $26 billion. We have negotiated exclusive rights to a first-in-class patent from Harvard Medical School that covers inactivation of Hippo in combination with chemotherapies. We filed our own broader PCT application that also covers the use of these molecules with and as immunotherapies, an exciting further route of exploration. And we have a proprietary drug discovery engine that allows us to rapidly identify and develop new Hippo inhibitors at lower cost than industry standards. This engine includes proprietary homology modeling, convolutional neural networks, biochemical assays, and now dozens of small molecule compounds that we are synthesizing, testing, and patenting. Leveraging this drug discovery platform, we were able, over the past 4 months at IndieBio, to develop our first Hippo inhibitors with just $178,000, compared to industry standards for similar work of over $2 million across 2 to 3 years of R&D. Nivien is not just developing one small molecule against one target for one type of cancer. Hippo contains 5 core components, and we have already developed multiple inhibitors against 2 of them to start. Once optimized, each of these compounds can be reformulated with any one of dozens of different chemotherapies to create a new frontline of care in every major cancer that outperforms the current standard of care in terms of both effectiveness and side effects. We are 18 months of development to get our existing inhibitors to the optimized point where they can enter IND-enabling trials, a key de-risking milestone prior to human studies. These optimized compounds will also serve as the foundation for an interim step in our business model, sub-licensing partnerships with pharmaceutical companies who have chemotherapies that are going off patent. These licensing partnerships will include an upfront payment, milestone payments, and a downstream royalty, enabling Nivien to begin generating revenue by the end of next year before we've entered any of our own clinical studies. We've spoken to 7 pharmaceutical companies and identified the specific metrics that our molecules need to meet in order to qualify for these partnerships. We are 18 months away from this commercial revenue-generating milestone. Nikita Shah and I founded Nivien as scientific researchers at Harvard and MIT, in partnership with the chair of the Systems Biology Department at Harvard Medical School, Dr. Mark Kirschner, who now also leads our scientific advisory board. We've recruited 2 exceptional scientists. Dr. Ken Fong is a principal chemist from Allergan, who led drug discovery projects there for 17 years, racking up 56 biochemical patents. Dr. Dharmendra Singh is a veteran cancer biologist from Houston Methodist, UCLA, and the NIH, with 18 peer-reviewed publications in cancer biology. In addition to Dr. Kirschner, our scientific advisory board includes the scientific founders of Moderna Therapeutics and Certris Pharmaceuticals, as well as a clinical trials investigator from Massachusetts General Hospital. Together, we are seeking to reduce the side effects of chemotherapy and make it a more effective treatment for cancer. We are raising $3.5 million to optimize our compounds to the commercial and developmental milestones that I've laid out. Our aim is to prolong and improve the lives of the millions of patients worldwide who rely on chemotherapy every year. I hope that some of you will join us in our journey to transform the standard of care in cancer therapeutics. Thank you very much.